Hi, so we are continuing our Photomicrobes lab tour, and here we have Emren, and he is also a PhD student, and he's going to tell us a bit about what he's doing here. So, could you explain a bit of what exactly do you do in the group, and what is your research project?
Hello, I'm Emren. I'm a third year PhD student. That since I'm working and living in Turku from November 2022. And I'm working here with the photosynthetic microbes group, as you said. Photosynthetic microbes group is a fundamental and applied photosynthesis lab. So in our lab, we are investigating to understand how the simple photosynthetic organisms like microalgae and cyanobacteria, how they are photosynthesizing, how they are harnessing energy from the sun, and how we can utilize this energy into producing algae biomass or the metabolites or products that we can extract and we can use in different sectors. So these are the questions in our lab. For my research, I am working in one EU project called relevance, the reusing the effluents from agriculture to unlock the potential of microalgae. I think it's a fancy abbreviation. And the most important aim of my study is about cultivating the algae at large scale and integrated this system with wastewater management in a way. So I want to produce biomass. Meanwhile, I want to treat the rainwater by using these green algae.
Well, see, that's very interesting. So the idea is to actually so have this combined agricultural wastewater treatment, so we can have like a greener agriculture, like greener in every sense, because the algae is also greener. But that's very cool. So this looks very cool. What is that? There's a bioreactor, but can you explain what is it doing exactly?
Yes, this fancy sci-fi setting is called the photobioreactor. That the photobioreactor also I can take.
Yes.
And photobioreactor, what we are doing, we are trying to create a system so our algae can always access the nutrients and light and the other gas, which is the carbon dioxide and air. So in the PBR, we are making sure that our algae is always running endlessly in those glass tubes and making sure that they are using the nutrients that is coming from the wastewater. So we are treating the wastewater as our algae grows in this photobioreactor, and we can harvest the algae and we can separate the treated water and algae in our filtration systems that I can show it to you later. So this machine enables us to grow algae in a very robust rate, let's say.
Yeah, cool. Yeah, I see there's a lot of the tube is like going around, so there's like quite a lot of like liquid inside. That's cool. And also there's a light source, as you can see, which is basically of course giving the energy to this algae. Very cool. More about the group, because that's a very interesting. How did you find about like the group, like to join it? When was it? Can you tell me a bit about the process?
Sure. After completing my master's studies, I was looking for PhD opportunities, and at the same time, luckily, the photosynthetic microbes group were looking for a researcher. So the researchers that they are looking for to cultivate microalgae at large scale with the integrated wastewater treatment project. So I saw their position announcement on website, and I applied it, and we interviewed it, and here I am now. So definitely like that.
Yeah, cool. In terms of technology readiness, how far do you think we are from actually integrating this into agriculture? Like, how many years would you, let's say, think that we're away from actually integrating into agriculture, into hydroponics?
Yeah, I can say that this is also called the bioremediation. So that is treating the wastewater and the rainwater by using microorganisms or any biological organism, let's say. And so it's not a new tactic to treat wastewater by using algae. In wastewater treatment facilities, they are always using a cocktail that is made of bacteria and algae. So they are also treating it. What makes this EU project different is to making sure that this system will integrate the greenhouses and the other agricultural facilities directly, and this will create a chance to treat wastewater and produce the biomass in a decentralized and autonomous way. In this type of system, we are making sure that throughout the year, we are creating a facility that we can produce biomass and we can treat the wastewater in the greenhouse facility or in any facility that is doing agricultural practice. So I think that makes this project special.
Wow, that's very interesting. That's truly interesting. And of course, if we can, let's say, how he said, decentralize and have this local wastewater treatment, imagine you don't have to care so much about carrying the wastewater like elsewhere to some, let's say, decontamination facility or, yeah, that's very interesting. You mentioned there is a bigger setup. Can you show us?
Sure, yes.
Cool.
Come then.
Hey, so this looks like, let's say, bigger, more complex, and like very cool version of also the previous system. What is the difference between those two, and why do you have this bigger version?
Actually, this bigger version is one of the deliverables that we have a validation plant in here in the University of Turku, and in our EU projects we are creating a recipe for let's say the private sector, and at the end we will say that to treat the hydroponic greenhouse effluent in a way that they can produce more algae biomass. They can use what we are doing and what we are validating in this PBR, and with that recipe our collaborators in the EU projects, they will use to grow algae in a bigger scale up to 100 meters square or 80 meter cubic volume level in the open ponds. So what we are doing is to make sure that we are trying to find a way to maximize biomass productivity and nutrient uptake performance of the algae. And with our recipe, our collaborators will use it. So we have a validation plan in here. But to inoculate in this 1000 liter PBR, you need something like 60 to 150 liters inoculum. Inoculum means the algae in a dense level. So we can use this smaller version of this PBR, which we showed before. Yeah. And we are inoculating our algae with the wastewater in the PBR, and we are making them run to make sure that our algae is literally eating everything inside the system while they are growing.
So you turned off the compressor now, but we can still see bubbles around. So these bubbles are actually injection of CO2, right, into the system.
For algae production, we need two main gases, which is pure carbon dioxide and air itself. That means in this photobioreactor, we have infrastructure to inject air and carbon dioxide whenever it's needed. Carbon dioxide we are using for two reasons. One is to regulate the pH. The another one is to make sure that since our algae is photosynthesizing, they need carbon. And the carbon is only coming from the carbon dioxide in that sense. But for the air is important to make sure that the system is mixed very well, and there is more technical details like the dissolved oxygen percentage that we need to regulate, and air is helping to make us do that.
Wow, very interesting, and so many parameters to take into account. Now thinking about, like, let's say a more after you apply and about like relation to other people and the world around us, how do you think your research impacts the world? Like,
right, the general question, but it's the most important question, because why we are doing those research in here. I am thinking about the importance, and I can say that the agricultural practices are the main reasons to why we are using the fresh water and the nutrients, because we need food as the population grows, and we will do the more agricultural practice. That means we will use more new nutrients like nitrogen and phosphorus that we were mining for them, and the fresh water, which those are limited sources. That means in the agricultural area, and whenever we are producing food, we need to find smart ways to use those nutrients and fresh water and keep them otherwise, the untreated wastewater that we are producing, and if we are not treating them well, if we are not controlling, they are disrupting the ecological balance. For example, eutrophication is a one problem that even we are seeing in the Baltic Sea, that if those nutrients are running through the marine systems and if they are reaching the coast, the algae that we are controllingly growing in here, they will uncontrollably grow in the coast or marine areas, and they will consume all the nutrients and oxygen, which we call this eutrophication, and that means they will create a dead zone, so all the marine ecosystem will disrupt, and for example, the fish will die. You will see in the marine ecosystem, which is quite disrupting. Plus, in the economical side, that the algae farmers and the greenhouses, that they are looking for the more economically feasible solutions, that because for each biomass production, there is a price in here, and think about it, if you don't need to use the different light systems, different heating and cooling operation to produce algae, since, think about it, this system will integrate in the greenhouse. And then in the operational sense, if they are not using any new fresh water and the new nutrients, and instead they are using the wastewater, you can see that how cheap we can produce algae biomass that would be a feedstock for different sectors. So I think this is the most important part in our research for the world, to make sure that agriculture and the algae production can go in a more sustainable and a more economical feasible way.
Wow, cool. This is truly a very beautiful project of integrating bio-circular economy, you see. So you're not only producing, for example, food, but you can also like treat this wastewater locally, and you can also have some also economic output from that and use this fresh water again while not generating residues. This is so cool. This is so cool.
May I add something?
Of course.
Because in this project, also we are doing a waste to product project, and it's a quite circular bioeconomy practice, I can say. Because we are using the wastewater to treat it. We are using wastewater to grow algae, and we are treating the wastewater by producing algae. So that means we are producing algae from wastewater, and we know that our algae has potential to be used as biostimulant and biofertilizer and pesticide effects, which they are high value added products. And those bioagrochemicals, they can be used in the same greenhouse. So you are seeing a waste to product opportunity. That's after this operation done in here, algae and the treated water can be used in the greenhouses again. So this is a nutrient recycle project also. So there are more than one way to achieve the sustainability, and this biotechnological solution that we are doing in here is offering this waste to product project, which is I think one of the most important aspects in our research.
What do you think of Turku and UTU as a place to do research, as a place to live? Do you like it?
Absolutely, because in the Turku I am feeling super safe first, so I am not worrying about my future in the near term, so I can focus my research very much, and the working community and the people around in the world and in the workspace, that they are quite helpful and respectful, which is quite important for me, because for example, I am working at the greenhouse and there are things that I need help for the stuff, and the Ruissalo Botanical Garden staff are helping me a lot, so I am quite thankful for them. And this applies to every department in the University of Turku. So by working here, I feel quite fine. So I can focus also on my research in those conditions, which is quite important.
Absolutely, absolutely. It's so important to have collaboration between people, and people with different expertises and different responsibilities, so everybody works together. That's very cool. I really want to see like those other cool things, but before we go there, could you tell, like, if, for example, if you were giving some advice to a master's student that is deciding either to come to University of Turku or another one, what would you say? Like, what would you tell them?
What I can say is to be a researcher and to study in Turku. Turku could be one of the best places to balance between the academic research and living in a peaceful and the respectful city, and still you can stay connected to the nature. So all those three, I think Turku could be a sweet spot for this. So I suggest to join to University of Turku team if they are planning to progress their academic career in here, in the master, in the PhD, in any position, because you will find a very supportive community, and that place will be full of opportunities for everyone that have a growth mindset.
Can we see those? Could you give me a bit of explanation?
Since we are at the greenhouse,
Yeah.
the wastewater is coming from this side, and in those canisters, we are storing the wastewater, and whenever we need, we are pumping the wastewater to our photobioreactor. I will say PBR afterwards. Before sending it, we have lots of compounds in wastewater, but we don't want to do so much pre-treatment. So you are seeing the filters, and we are only filtering it. We are not doing anything else. And then this filtered wastewater, you will see a pipeline through the treated wastewater is going to the photobioreactor. And our algae from the other compartment joins too. So this is called the inoculation process. And as our algae is joining to PBR, our PBR is turning into green. That means our algae is growing. And every day we have on-site probes. And we are taking a sample and we are measuring lots of variables at the lab to understand how fast our algae are growing and how fast we are treating the wastewater. So what is our nutrient uptake performance in this sense? When it comes to a time, it's time to harvest our algae and harvest actually the treated rainwater. So what we are doing, that we are adding more wastewater to the system and we are elevating the water level in the vessel. And from there, it's connected to canister, and with that canister, we are filtering the algae culture. So this is a filter. So with this filter enabling us to separate the treated rainwater and the algae. So we are getting a concentrated algae at the end from this filtration unit, and the wastewater we are collecting in different canisters.
They're recycled. Yeah.
So these are we are saying the recycled or recirculated rainwater, and we are taking a sample from it, and we are checking that what is their current nitrogen level, what is their current phosphorus level, what is their pH, and then there is a thing. If we are seeing that nitrogen and phosphorus levels are not below regulatory level, we cannot discharge the rainwater. So we need to pump this recirculated water back to PBR again. With the fresh wastewater and the recirculated wastewater, we are combining them and we are pumping the system again. And also in that method, we are controlling how much nutrients we are giving to system, because we know how much our algae are consuming the nutrients. So we are balancing it and we are making sure that we are doing a successful wastewater management. We are keeping the concentration level below regulation level, so we can discharge at the end, but we don't want to discharge it. We want to keep it and we want to maybe send it back to the greenhouses again. So greenhouses can use a similar water. So they don't need to use any additional fresh water in that sense, plus the nutrients.
Wow.
In that logic. And then we are storing our algae in freezers, and we know that our algae has lots of compounds inside, some fatty acids that we can use, for example, omega-3, omega-6, they are fatty acids, and we can use those fatty acids in pharmaceutical sector and also in the aquafeed sector. So, which means literally we can feed our fish with algae, because it's happening in the nature again. The planktons are also algae, and they are eating the algae to get those omega-6 and 3s. But specifically for my project, I am with the team, we are checking the biostimulant and the biofertilizer and biopesticides potential of algae, and we are seeing that it works. If you are applying this algae to the plants, our plants are growing better or they are healthy, we are enhancing their features. So what does it mean? From the greenhouse wastewater, we created an algae as a biofertilizer and biostimulant and biopesticide, and we are able to use it back to the greenhouse again. So this is our story in this compartment.
Wow. Quite a plentiful of different opportunities in the algae. So as a final thing, could you complete this sentence, get inspired by? We get inspired by science and at UTU in general, but do you have a specific inspiration? Get inspired by what?
Yeah, since this project and since my department is mostly also thanks composed of the biological operations with the help of the machines and some technological applications to maximize or unlock their potential, I can say that I am getting inspired by biotechnology. So this could be a sense.
Very cool. Thank you so much. I'm afraid our time is running out a bit, but thank you so much. See you in the next video.
Yep.